Hi, what are you studying?
We are studying methods of gene transfer.
We are also not able to understand the methods of gene transfer.
Can we study together?
Let's study together the methods of gene transfer.
What is gene transfer? Gene transfer is the introduction of foreign gene artificially into an organism by overcoming the fertility barriers. This process is also known as genetic transformation, and it is a very important step in genetic engineering. Different methods are available to achieve genetic transformation of organism, that is, the delivery of foreign DNA into the host organism. These are divided into two main groups: direct methods and indirect methods. Indirect methods, in this case, vector is needed for insertion of the foreign DNA into the host genome, whereas in direct method, the vector is independent. The DNA is directly inserted into the host genome. Indirect methods include Agrobacterium mediated gene transfer, and direct method is further classified into physical and chemical method. Physical method include electroporation, heat shock, microinjection, macroinjection, liquefaction, and ultrasonication. Whereas chemical method include calcium chloride and PEG mediated gene transfer. Agrobacterium mediated genetic transformation. This method uses Agrobacterium as a vector to introduce the gene into the host cell for the plant transformation experiment. In this method, Agrobacterium tumefaciae bacterium is taken having a Ti plasmid. The Ti plasmid consists of tDNA. This Ti plasmid is removed from the bacteria. The tDNA is cut by the restriction enzyme, and the target DNA is also cut by the same restriction enzyme. The target DNA is inserted into the tDNA of the plasmid, leading to the formation of the recombinant plasmid. The plasmid is introduced into the bacteria. The transfer of tDNA carrying foreign gene into the chromosome of the plant cell. The plant cell are grown in culture. There is a generation of plant from a cell clone carrying the foreign gene and may express it as a new trait. The plant is called as transgenic plant.
The heat shock method is a bacterial transformation technique that facilitates the uptake of DNA through temporary pores in the bacterial cell wall. The procedure is as follows. Mix chemically competent bacteria and DNA in ice for a short time. Place the mixture at 42 degrees Celsius for 45 seconds. The heat shock interval depends on the surface to volume ratio of the cell suspension. Place the heat shocked cells back in ice for at least 2 minutes. Add SOC media or LB and incubate the transformed cells at 37 degrees Celsius for 30 minutes with agitation. Play 2 quantities of transformed bacteria to ensure isolation of colonies.
Electroporation. Electroporation is a method used in gene transfer where brief electrical pulses are applied to the cells temporarily, increasing their permeability and allowing the introduction of foreign genetic material into the cells. Electroporation is basically done in 4 steps. First, the cells are suspended in a conductive buffer containing the genetic material to be introduced. Second, the cell suspension is placed between 2 electrodes. Third, a brief high voltage electric pulse is applied, causing temporary pores to form in the cell membrane. Fourth, the genetic material enters the cell through these pores. After electroporation, cells are typically allowed to recover in a suitable growth medium.
Microinjection is one of the physical methods of gene transfer used to introduce DNA or other genetic material directly into a cell using a small glass needle or micropipette. In microinjection, firstly the glass micropipette or needle is prepared by heating and stretching the glass to form a fine tip. At the heated end, the resulting tip size is usually around 0.5 mm in diameter, resembling an injection needle. The entire process of delivering foreign materials using microinjection is performed under a powerful microscope for precise manipulation and observation. The cells to be microinjected are placed in a container. A holding pipette is placed near the target cell. It uses gentle suction to hold the cell during the injection process. The micropipette containing the desired contents is mounted on a micromanipulator. which allow precise positioning and movement of the pipette. The micropipette is lowered near the cell. The micropipette is carefully inserted into the cell, either into the cytoplasm or the nucleus, depending on the target location for the genetic material. The contents of the micropipette are then injected into the cell by applying hydrostatic pressure using a microinjector. The pressure forces the fluid containing the genetic material to be released through the micropipette into the cell. After delivering the content from the cell, the empty micropipette needle is slowly and carefully withdrawn from the cell, minimizing damage to the cell and its membrane. Proper technique and high quality micropipettes helps minimize cell death during this step. Macroinjection is the method tried for artificial DNA transfer to cereal plants that show inability to regenerate and develop into whole plants from cultured cells. Needles used for injecting DNA are with the diameter greater than the cell diameter. DNA injected with conventional syringe into region of plant with will develop into floral tillers.
Lipoification. This is a lipid mediated transfection technique which is used to inject genetic material into a cell by means of liposome. Liposome are a microscopic vesicle that can easily merge with the cell membrane since they both are made up of phospholipid bilayer. As you see in the diagram, we have a lipid solution that is positive charge and the DNA having negative charge. We mix them up both. After mixing, DNA is encapsulated in a lipid bilayer. After that, we added into a cell. Liposome adhere to the cell by the process of endocytosis. After that, the liposome fuses with the plasma membrane of the cell and the DNA enters it. As a result, DNA reaches to the cell. Ultrasonication. This formation of a small pores in a cell membrane using ultrasound allow the transfer of DNA, RNA into the cell. This phenomena is known as sonoporation and is a gene delivery method that shows great promise as a potential of new approach in a gene therapy.
The calcium chloride method is a commonly used technique for gene transfer that introducing foreign gene DNA into bacterial cells. Generally, in this method, bacteria are treated with calcium chloride to make their cell wall permeable, that allowing the uptake of foreign gene DNA. The DNA of interest, typically a plasmid, which containing the gene of interest, that mix with the bacteria and calcium chloride solution. Then heat shock is applied to the mixture that promotes the uptake of the foreign gene DNA by the bacteria. Page mediated method is a technique that used in gene transfer, particularly in cell culture or in vivo experiment. It involves the use of page to fuse cell membranes, that allowing the transfer of genetic material such as plasmid DNA or viral vectors into the targeted cells. Page helps overcome the barrier posed by the cell membrane, that facilitating the introduction of foreign gene DNA into the cells.